My name is Olivia Vorius. It is 7/6/25. I'm doing my project on the prokaryotic and eukaryotic cell interaction, which I'm starting now. All right. Let's see. So prokaryotic and eukaryotic cell interaction. How do prokaryotic gut bacteria interact with the cells of our GI tract? First, we have where this occurs. So it takes place in the intestines where we have our microbiome, and the two bacteria that I highlighted were lactobacillus and positive E. coli strains. So the basics. Helpful gut bacteria such as lactobacillus and beneficial strains of E. coli populate our gut microbiome and interact at the level of individual enterocytes, aka intestinal cells. In my diagram here, I drew the actual villi itself, comprised of enterocytes and the lactobacillus and E. coli making their way through the villi and into the actual gut microbiome itself. The blue layer is the mucosal layer. So to go into more detail, how does this symbiotic relationship actually take place? Number one, positive gut bacteria are introduced during and shortly after birth via breastfeeding and in the diet as adults. Helpful bacteria cells are recognized by the enterocyte pattern recognition receptors, while surface layer proteins of the bacterial cells help adhere the bacteria to the enterocyte itself. So even in that sense, the bacterial cell and the individual enterocyte are working together. Next, bacteria then form a symbiotic relationship to the enterocyte by helping produce mucus that can trap harmful pathogens as well as maintain strong barrier integrity. More on that a little bit later. Even the simple act of good bacteria adhering to the enterocytes help them outcompete harmful bacteria by taking up space and not allowing them as many cells to bind to. Last, enterocytes help the proliferation and support of good gut bacteria by absorbing nutrients and thus producing substances the microbiome can utilize for energy. In another diagram, I did a little bit of a closer look of the lactobacillus actually adhering to an enterocyte itself. So I labeled the mucus layer again, I labeled the lactobacillus bacteria and where it bacterially adheres to the enterocyte. The microvilli are also just tinier versions of the villi on the surface of the individual enterocytes. And then the two sticks together, pointing to the arrow that says cell surface proteins allow bacterial cell adhesion. That just shows you how the cell itself has proteins that interact with the bacterial cell. And for reference, enterocytes are significantly larger than bacterial cells. Now I want to discuss the immunologic relationship. The villi hold the largest population of immune cells in the body. More specifically, they contain Peyer's patches. These hold lymphoid tissue and tolerate our food, and most important, our beneficial gut bacteria. However, they do also respond to bad bacteria. For example, Salmonella bacteria enter the GI tract. Then, sampling antigens on the Peyer's patches of the gut mucosal cells alert the gut to begin an anti-inflammatory response. This results in dendritic cells proliferating T cells, which begin a cascade of key inflammatory molecules. Phagocytes and inflammatory cells attack the pathogen in order to mitigate disease. So how does this affect us on a larger scale? The gut microbiome relationship with the cells of the intestinal tract provide multiple benefits to us. The ability for enterocytes to feed and support gut bacteria allows further proliferation and improved immunity, nutrient absorption, and there are even connections to a healthy microbiome and mood improvement. To elaborate on this, the vagus nerve is a nerve that runs through the GI tract and responds to neurotransmitters produced by gut bacteria. These neurotransmitters, such as serotonin and GABA, promote a positive mood. So even on the cellular level, gut bacteria and intestinal cells work together in ways that have a huge impact on our overall experience of life. All the more reason to eat well, sleep, and manage stress. All of these factors benefit the microbiome and keep it functioning smoothly.